So what are we proposing to do with eDNA, and why are we doing this? Obviously, as lay people, we're just trying to support information gathering about the Little Miami River, and particularly about diversity, because diversity is a good thing. And it doesn't take much logic to realize that, because the food chain is very important. It's a very important part of the ecosystem. And if you have a lot of diversity, that means there's a lot of food for the animals that live there. And it's an indication of a healthy stream. So what we are doing is collecting water samples. That's all. There's no in-laboratory science or anything on our part. But in order for the diversity to be documented for the many different species in the river, we have to collect water samples. And we have a program where we are sampling during the current biannual period of some grants that we have. We're sampling 50 sites on the Little Miami River to collect water samples and to properly prepare them for sending on to a laboratory so that they can tell us, and we know that this works because we've been experimenting with it, they can tell us every species of fish population that is indicated to be living there based on the eDNA samples that we have sent to them for analysis. Again, diversity is a key indicator of health in the food chain and a healthy stream, and that's what we're out to determine, the extent to which we find a healthy diversity of fish in the Little Miami River. Now, it's amazing how simple this process is. And the reason it's so simple is because the technology has roared forward to improve what we can do with each sample. So all we need to do is to collect a 60 cubic centimeter syringe of stream water or more and force it through a filter that is screwed onto the syringe. And in the picture here, that's what's going on. That's my friend Dave Smith. He's got a syringe there, and he's going to pull in 60 cubic centimeters of stream water, and that's the sample that he gets. Now, the instructions on the collection materials say that if you very easily push 60 cubic centimeters through the filter, then do it again. Take the filter off, get another 60 cubic centimeters. And another if you can, so that by the time it almost clogs completely, you have filled it up with DNA evidence that will be searched out and analyzed at the laboratory. This is a picture of my friend Dave screwing the filter on to the syringe, and this is the filter housing. The filter is inside of this, and when Dave pushes down on the plunger here, you see water come out the other end, but the DNA evidence is collected in the filter. So then once you have that, you have a filter which is clogged theoretically with eDNA. Then you want to preserve that. So the next step is that there's another syringe. You unscrew the housing from the big syringe and you attach it, again, screws in right here to this smaller syringe, and it's preloaded with preservative material. So there you can see it. It's now, all the preservative has been run through the filter, and it's time to cap it. And these red caps, which are supplied with the kit, you simply screw the red cap in on one end and then unscrew it from the syringe and screw it in on the other end. And there you have a completely capped and preserved eDNA sample ready to prepare and send in the mail to the laboratory. This is another picture of the capping procedure. You can see the threads in here, and these threads fit both the 60 cubic centimeter syringe and the preservative syringe. So Okay, then once you're capped, you're all set. You're not going to contaminate anything on the inside. And this black envelope here, it's a plastic envelope, is provided with a barcode to house the sample. So you put the filter sample with capped inside the envelope and head for the post office almost. The kit contains a blue mailing pouch, and you put the black syringe envelope into or filter envelope into the blue mailing pouch, and it's prepaid, ready to go, first class package to the laboratory whose address is here. And we put a return address on here, and then she's ready to go. Here's a package that I sent in. I don't remember when that one was or which one it was, but all these barcodes on this material protect the identity and enable us to retrieve the sample, track it with UPS or with mail, first class mail tracking, and so on. So we've done this actually. We wanted to experiment with this system and see how it worked and what kind of results we got. And to do that, we partnered with the Cincinnati Nature Center at their Long Branch Farm area, where a mile of, I think it's approximately a mile of Long Branch Creek flows through their property. And this is the director of their Center for Conservation and Research. He and I went down there and I demonstrated this sample taking process for him, and he was really pleased. And we've also put in a water quality, or rather a water chemistry sampling once monthly over the summer as part of the Green Acres program, the stream snapshot. So that's really what you have to do. Now, one thing I want to point out, in order to submit a sample, you have to have an account. And it seemed madness for us to ask our volunteers each to open an account and go through the computer stuff. And we would probably have a hard time keeping track of it. So we have a Little Miami Conservancy account, and we will do all the packaging and mailing. That way we know where everything is. And we've had great communications and support from Noah Ventures, who is the creator of this system and provides both the packaging and the collection equipment, and also the laboratory processing. All right, so what does this have to do with freshwater mussels? It's really an interesting time we're living in, because this technology here is racing forward so fast that it's hard for us to keep up with it. There are so many academic papers claiming the wonderful things that eDNA will do, but a lot of it has not been proven yet. And one of the reasons it hasn't been proven yet is that in order for an eDNA sample to be analyzed by a laboratory and what they call sequenced, which is the process by which they determine the exact identity of the species there. In order for that to happen, the lab has to have a library of the fish and also the mussels. Well, the fish library is complete, and they have so many species of fish, some saltwater, some freshwater, that they do a thorough job of identifying many different species, and we're getting good results from them. However, the same is not true of freshwater mussels. It's a lot more difficult because freshwater mussels bury themselves in the substrate, they hide, they lay on the bottom, and so it's difficult, and they're also heavily protected because of their declining numbers in both species and individuals. So until a really good and diverse and broad library of freshwater mussels is a part of the DNA process and possessed by the laboratory until they have a freshwater mussel assay, they call it, in their library. They're not publicly testing yet for freshwater mussels. However, they can retest samples submitted that are stored in refrigeration. And so we're hoping, and also we've been working with them to help them develop the library for freshwater mussels. We're not sending them any tissue or anything, but we're trying to identify what freshwater mussels are in the Little Miami River system. And with the help of previous work that has been done by Michael Horgoth and others, we know what species should be there. We just don't have the library yet to prove that, because it's not complete. But the predictions are that they're going to have it this fall, or at least by the end of the year, and we're hopeful that that can happen. Okay, so another thing we're doing with the eDNA is we're finding the diversity of fish species, which is very important, because reproduction by freshwater mussels in almost all species is dependent upon a host fish taking the embryos from the female freshwater mussel and nourishing them for anywhere from a month to 2 months, and then releasing them as baby mussels. And so if there aren't sufficient host fish, there aren't going to be sufficient mussels. So what we're doing is we're trying to identify potential host fish in the reports that we're getting on the 50 sites that we're testing between now and June the 30th of 2022. This is a potential host fish that was caught. You can see a tiny nymph there on its nose. That host fish was caught at the same location as our potential host fish, at the same location as the water sample for eDNA was taken on the Long Branch farm. So that at least tells us there are potential host fish there. Now, this is a picture that was taken, I believe, by Monty McGregor, who is doing research in Kentucky. And this shows how this is an endangered pink mucket. It shows how the animal buries itself, half buries itself in the substrate, in the bottom of the stream. And so there's only a little of it showing. This is what the shells look like. And so it's no wonder that it's difficult to locate them. And when you find one, you'd better leave it alone, because it's protected and you're not allowed to remove a freshwater mussel from the stream. Okay, here's another tiny fish, which is a potential host fish caught again on a small insect on a fly rod. And another one. Now, this is a 2-page exhibit that shows how we're working out the collection system here. We have selected, actually, on this exhibit. This is page 2 of the exhibit. No, team 1. Try to go to team 2. I think there are 48 or 49 sites, and we're calling it 50 because frequently we'll see a new site and we'll want to sample there. So, and what we've done here is we have divided these up into manageable areas and created teams. And in this left-hand column in the yellow, you have collection team 1, team 2, team 3, and so on. This is the confluence with the Ohio River, and we work our way up. You can recognize some of these areas here, but we also have coordinates, these locations of these sites precisely, so that you could put the GPS coordinates in and go right to the site. What we're hoping for is to have all of these teams filled with trained collectors. Here we have Greg Schran and Jim Farsing. Waldorf Schools has this one, and here we have an open team. When it says team open, that means we're looking for volunteers who will be willing to do the sampling there during the next year and a half. And we go all the way to team 14, and then we have a tributary here, which is Jacoby Road Access and John Bryant State Park. We should probably call that team 15. But we're going to make this information available and try to get teams ready to help us collect the samples for all these places. Now, here are some slides from the Jonah Ventures educational material that again explains DNA from a little different perspective. Just imagine, I mean, people say, well, how do you get the DNA from a fish? If you've ever had a goldfish and forgot to change the water overnight, you know it just fills up with excrement, because the fish, if they're eating, they're gonna poop, and that will dissolve in the water, and it will be loaded with DNA. And that's what happens. That's how DNA gets in the water, mainly through excrement. There are also, during spawning season, eggs and so on. So what comes from the organism is really environmental DNA, and it's everywhere. A traditional sampling for species usually requires a team of people, and sampling, locating fish. We tried to do it with traditional fishing methods, and we also used some electrofishing. But this sampling needs only one trained person to actually do the sampling. We like teams to be formed for safety purposes. But also it can be done without touching, seeing, or in any way harassing or disturbing the animal, which is wonderful, because even though fishing is an accepted thing to do, and electrofishing is legal if you have a permit, it's really not quite as kind as simply collecting DNA. There's no harm at all to the animal for that. And another thing is it detects the presence of species that are sought, invasive, rare, or dangerous, and it can be used to estimate the relative abundance of species, the biodiversity, how many species live in the ecosystem. So that's basically it. The hope is long-term that we will learn so much about our diversity in the Little Miami River, and also the presence of freshwater mussels, that we will be able to join with researchers in selecting a site or 2 sites or 3 sites on the Little Miami River where freshwater mussels can be reintroduced after being cultured in the lab. We know that in vitro fertilization can be used, and that eliminates the need for a host fish. And baby mussels can be raised and released. But we know that not every inch or mile of the Little Miami River will have the best place in which to nurture them. And so we want to be in the position to make wise decisions, or to provide information to people who are making the decisions about where reintroduction of the species can be achieved. That's a dream, but it's not a dream to want to know what our fish species looks like, and to do the sampling over 50 sites on the whole river. We're very proud of that. We've got grant support for that, and we're looking for people to join us in helping achieve that. So I think that's all that I have. Anybody have any questions, Eric or others?
Bill, many thanks for walking through the process and how we're approaching this issue. I think obviously the more we know about what is living in the Little Miami, the better, and it will help us in the long term with strategies and looking at trends. If we do these mussel eDNA and even fish host DNA samplings, that this data done consistently year in and year out we think will start to show trends in the health of the overall health of the river. So a worthy effort, and we appreciate your work, Bill, and also certainly the funding that has come our way, courtesy of the Department of Natural Resources Division of Wildlife, in making these activities, this effort possible. So thanks again. It was a very informative session.
You're welcome, and let's go to the river and get going.